From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on August 15, 2024. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 54th episode of Matters Microbial. Thanks so much for listening and/or watching and for spreading, as always, the good microbial word. It's been a fine summer for my Doc Martian undergraduate research students, and mysteries still abound, such as work Hannah has done in my laboratory investigating what appear to be huge bacterial filaments in a local high-sulfide seep. I cannot wait to see what she finds out during the coming year in lab. It's really a pleasure. The 50th episode pins for Matters Microbial turned out quite well, as you can see. As I start to gear up to teach my fall semester microbiology course to some new Micronauts, I remind myself with this keychain of what a student called me many years ago, a microbial badass. I'll try my best to live up to that high praise, as Nicolas Cage would say. Here is the official logo for Fall 2024 Microbiology as we explore strange new microbial worlds. Artwork is by the wonderful Rachel Webster. Students will receive this image as a sticker, a button, and later a t-shirt. I have done this for many years now for my students, and it's all about the microbial PR. The term microbe itself seems to suggest smallness. As we've learned in an earlier episode of this podcast, this is not always true. And viruses are usually thought to be still smaller than most bacteria and archaea. But once again, the microbial world is, to borrow from JBS Haldane, stranger than we can imagine. Here is a comparison of the sizes of some of these giant viruses compared to a bacterial cell. The first of these giant viruses, termed a mimivirus, can be seen here in a 3D print that my wife, Dr. Jennifer Quinn, printed from an NIH file. The more scientists investigate giant viruses and the effects they have on their hosts, the greater the mysteries unfold. I will be adding many fascinating links to today's show notes for your microbial enjoyment, I can promise you. Because I love giant viruses and want to learn more, I'm especially delighted to welcome Dr. Mohammad Moniruzzaman of the Rosenstiel School of Marine, Atmospheric, and Earth Science at the University of Miami. Dr. Moniruzzaman will tell us about the work his research group has done investigating these colossal giant viruses and how they have evolutionarily shaped eukaryotes in marine environments. Monir, welcome to the Quality Quorum today.
Hi, Mark. Thank you for inviting me. It's a pleasure to meet you on this quorum, and I'm very excited to talk about giant viruses to the audience today. Yep.
I cannot tell you how exciting this is for me. I teach about it just a tiny bit. Remember, I just have one microbiology course at this small undergraduate institution, so I have to see how much I can pack into that really, really complicated coursework. But I never forget to talk about giant viruses. Although I believe you have corrected me in the past, people sometimes use the term gyrus for giant virus, but this is a no-no among people who study it.
We are not super excited about that term, but we know that people have used this historically, and I think that term is more like affectionate kind of way of saying, referring to giant viruses, but it does not really reveal any information that's useful. So particularly giant virus itself, I think it's a sweet and simple term, so we can use them all the time. So I am more in favor of giant viruses instead of gyrus, whatever you call them.
I think it's a really good way to describe it. Is just say use the term. Now, most people listening or watching are used to the idea of viruses being quite small, and in fact, some of them are. But these giant viruses are fascinating, and I thought if you could, can you give us a short introduction to them?
So yes, definitely. I think one thing that I want to clear up a little bit is that giant viruses are big, but within that whole family of viruses, or I should say, whole domain of viruses, there are quite a bit of variation in size. Like you do have those lilliputs in the giant family, like the smaller ones, and then you have these massive ones, which we'll get to, eventually. But to give a brief introduction of giant viruses, I'm assuming that audience knows what viruses are, but I would just tell you that historically viruses are thought to be much, much smaller than bacteria. A bacteria we can see under a regular microscope nowadays, which we call bright field microscope. Viruses are not, you cannot see viruses under microscope. When I say viruses, the regular viruses. Giant viruses are technically, what you can say is that some of the giant viruses, you can actually observe them under regular microscope. And here is a picture, or I would say more like a funny illustration, where you can see some of the giant virus members along with E. coli. E. coli is a bacteria, and the illustration was drawn by my brother. And you can see that, yes, you can see that it's kind of a caricature, or like it's referring to the Beatles' walk in London. So it's roughly in scale the sizes of the viruses. So you can see that some of these viruses are almost as big as E. coli bacteria. So these are massive viruses, and they infect. So basically, their hosts are eukaryotic cells. It can be multicellular animals, for example, fish. Some fish have giant viruses that infect them. But vast majority of these giant viruses infect single-celled eukaryotes, which we call protists. And they're very common in freshwater and marine environment, and even in soil. So we do see a lot of giant virus signatures in marine, freshwater, and soil environment so far. So that's a basic introduction. When it comes to a comparison to regular viruses, a simple example could be the SARS-CoV virus that we have been going through right now, the pandemic. A regular giant virus can be, I don't know, like anywhere from 10 to 20 times larger than a SARS virus. And I say that because SARS is actually considered a large virus in the virus RNA virus realm. So SARS is actually a big virus. Compared to SARS, giant viruses can be anywhere from 10 to 20 times, or probably even larger. I don't have all the stats in my head right now, but just to give a basic reference to the scale. So yeah, that's the introduction.
So for the introductory micronauts, people new to this kind of thinking that we try to do on the podcast, can you address what advantage a virus could have for being so very large?
That's a good question. And again, this is also a question that scientists are trying to answer. Like what is the benefit of what we call this? We call this genomic gigantism. So what is the benefit of this gigantism, right? One idea which, and again, like many things in science, there might not be a straightforward answer to this. But one of the possibility that giant viruses having these giant genomes is that it gives them, number one, some kind of metabolic independence. So they don't have to rely on host machinery as much as some of the smaller viruses. Because, one of the response of the host is to shut down its metabolism when it gets infected by a virus. It's trying to prevent the community. It doesn't want to get the virus propagated and infect new cells. So if the virus brings some of these genes with it, it might actually help it to actually overcome some of that shutdown, avoid that disaster, that it can actually keep going even though the host is not cooperating. Another thing which could be tied to this same idea is that it also can help them switch their host. It's like, switch. If you have a Swiss Army knife, it can help you to accomplish many things. if a virus is infecting very different type of host, those two hosts have different metabolic environment, then having many genes might help them actually adapt to this new environment. So, one example that I can give you is that amoeba can be infected by multiple types of giant viruses. They are very distinct, even though they share an ancestor. These viruses are sharing a common ancestor in the evolution. Those viruses have very distinct history. Like, they have diverged like millions of years ago from each other, but they're still infecting the same host. So you can think about all these viruses having so much different metabolic capacity. Infecting the same host means that those viruses have to adapt to this similar host environment. And having all these genes might actually help them to adapt to the new host that they're infecting. So these are my thoughts, and none of these are really kind of validated, but I feel like these are good hypotheses to start working on. Yeah.
It's interesting to me, and I don't want to throw anything off, but when we talk about a virus, we often think about like the genetic information enclosed in a type of a packet. And that's the way that you can think of things, starting with, say, tobacco mosaic virus, which is one of the simplest we can think of. And at the same time, when we look at these giant viruses you're talking about, they have enormous amounts of DNA compared to the viruses that we're used to. And this is such a mystery, and I hope that later in your talk you can touch on that issue.
Yes, I definitely want to touch on that. I think that's a very fascinating aspect of giant virus biology, and which we study in the lab to some degree. Giant viruses do have massive genomes, which is kind of goes with their massive size. Some of these viruses do have genes. Like if you say that a virus usually do not encode many genes, anywhere from, I would say, 2 to maybe 20, 30 genes in a regular virus. But giant viruses, sometimes they can encode up to 1,000 or more genes in one single genome, which makes them a very complex entity to study because, number one, most of these genes, we do not know what the functions are. But if whatever we have known so far, there are genes in giant viruses that are involved in cellular metabolism. So when I say that, it means that a regular cell, like a human body or any bacteria or any kind of thing we consider living, have genes for metabolism. For example, we have carbon metabolism in our body, amino acid, or digesting food. So we're producing enzymes. So there are like all these genes that produce those enzymes or contribute to energy transfer and energy generation in the body. So it could be glycolysis, for example, the classic TCA cycle, where we're producing ATP or energy. So these genes, these metabolic genes, we call them, are usually not found in viruses because, as we know, viruses are really dependent on their host machinery to propagate. What we have seen in giant viruses, especially in the marine realm, there are some giant viruses that encode genes for fundamental biology. For example, glycolysis or Krebs cycle or TCA cycle that we call it. Pathways that are fundamental for energy generation, ATP production in any cellular being. Giant viruses encode those genes. So that's one thing that I just want to touch on, that they have this complex, almost like their own metabolism. But we know they're virus; they don't really replicate without a host. So why are they metabolically kind of partly independent? We still are answering that question, but it makes them fascinating to study. Yes.
I'm really glad you brought that up, because one of the important things that I want listeners and viewers to appreciate is that when viruses are outside of a host cell, they are not expending energy. So they have no innate metabolism. Now, I have studied for some years, in my way anyway, at a small undergraduate institution, a bacterial predator that in many ways seems like a bacteriophage in the fact that it attaches to a host organism, it takes it over, turns it into a factory for making more bacteria. People might think of that like a bacteriophage, but in fact, when Delovibrio, this organism, is outside of the host cell, it's expending DNA, it's expending ATP, it has an active metabolism. So I keep cringing, waiting for the day that we find out that one of these giant viruses actually has some metabolic activity outside the cell, because I've been saying that no virus does this for so long. But that's how microbiology is.
Yes, and we fundamentally, so for Delovibrio, it's a parasitic bacteria, which is pretty well known and well researched. But when it comes to giant viruses, the fundamental difference is that by definition, a virus cannot be metabolically active outside the host. Or by definition, a virus is not going to replicate by itself. So what we see, if we see a giant virus, like you said, that it's metabolically active, will you call it a virus? That's the question, right? It might be called a living being, and we have to rethink lots of our evolutionary kind of framework that we use to describe biological entities, right? So, I mean, you're right that if we see something like that, that's kind of on the verge of, I don't know, like, yeah, I think then that I cannot call that a virus technically. I have to redefine that. I have to find a place on the tree of life for that particular entity, but it won't be a virus,
No, I think it's a lovely philosophical discussion. So other than the size of the virus and the fact that it's carrying such a large amount of DNA and often metabolic genes, are there other characteristics that make giant viruses different than other viruses?
Yes. So one of the very prominent feature, and again, this is not all giant viruses. Some giant viruses have this really interesting components on their surface, which we call like fibrils or fibers. So that like almost like beard-like or hair-like fibers all over the capsid. Yes, kind of like that. Yes. So that's, for example, the Mimi virus has that those kind of features on it. When it comes to other features, giant viruses routinely they can pack proteins, not just DNA. They can actually pack a lot of different type of enzymes and other proteins that can actually help in the initial stage of the infection. So they carry those proteins with them, along with their DNA, and they can use those proteins when they start infecting to do some like initial terraforming, as you say. Like they can probably use those proteins to modify the host environment to some degree that's more suitable for the infection.
Just as, for example, retroviruses will carry reverse transcriptase. I tell the students it's like packing a lunch.
Yes, it is. It is. They're packing it. And I'm pretty sure. And again, here I could probably talk about a paper that recently came out. Giant viruses can have so many fascinating type of shapes and sizes, and so many appendages. Some of them have this like weird tail-like appendage and all kind of stuff.
But that takes us away from your work, and perhaps you want to explain what your laboratory is studying and the fun you've been having.
Yeah, well, we have been having a lot of fun. So I started as an assistant professor at the University of Miami's Rosenstiel Marine School in 2022. So, I started, the lab was empty, I was new and lots of things to figure out as a new faculty. But eventually I recruited some really good students and, we built a small team. We have 3 PhD students now, 1 master's student, and a number of really enthusiastic undergrads. So basically, we are really exploring the giant viruses in the marine realm. So one of the biggest questions that I have is, and we touched on this a little bit before, is that the metabolic genes that these giant viruses have, how do that contribute to the success of these viruses in the marine ecosystem? Like, why do they? So one of the biggest simple concept in evolution is that an organism or even a virus will not keep things that's not useful. So if a virus is keeping these metabolic genes for so long, in the evolutionary time scale, they must provide some benefit to the virus. And one of the biggest questions that we have is how are these metabolic genes are contributing to their success or persistence in the environment? So we have a couple of systems in the lab, which is basically a virus and a host. Basically, we have a single-celled protist host and a giant virus that infects it, which is marine. And I want to show this picture to you. This is called Rosenstiel. Rosenstiel is a mixotrophic protist. A mixotroph is that a virus, a host, that can actually thrive both on photosynthesis and it can also consume bacteria. So it has two modes of nutrition, primary production and also heterotrophy. That is, it's actually can graze on bacteria and it can survive. So this Rosenstiel, this mixotrophic algae that we have, it is also infected by one of the largest giant virus that infects an algae. So the picture that I'm showing you here, you can see that this virus is actually coming out of the Rosenstiel cell. It's a lovely SEM picture that we have. So yes, so these are massive viruses with a lot of metabolic genes. This virus, we know that has genes involved in central carbon metabolism, TSA cycle, and glycolysis. So right now, one of my students, he is working on understanding the role of these genes in the metabolic success of the virus during reproduction. Yes, that's one thing. We also do field work. We go to the environment and collect the community signatures of giant viruses. We do DNA and RNA sequencing. So we're funded by National Science Foundation to do some work on actually looking at field signatures of giant viruses and how this community changes over time. So one of the biggest questions that we have is, are all the families of giant viruses equally successful? Like, do we see some of the families more abundant in particular regions of the ocean compared to others? And that will also tell us a lot about what kind of host they're infecting and what impact they have in the biogeochemical cycle in the ocean. The giant viruses are so common in the ocean, they must have a significant biogeochemical impact. They must be killing a lot of these microbial cells in the ocean and producing these viral signatures and turning over carbon and other nutrients. And we want to understand that processes a little bit more clearly. So briefly, basically, our lab component to understand the genomic underpinning of their metabolic success, and a field component to understand better their ecology in the marine ecosystem. That's what we're doing right now as we speak.
So where do you go to do the field work?
So mostly coastal. So far, mostly coastal work. We're doing one thing that we have learned that in some of the coastal ecosystems, these viruses really thrive because coastal systems are generally productive. So lots of nutrient input, lots of phytoplankton blooms, and phytoplankton growth. So the viruses are actually following this phytoplankton or protists. And in the open ocean, they're obviously there. But in my opinion, it's an opinion because we don't completely know the answer, that they can be quite common and they can be more abundant in the coastal systems compared to the open ocean. So far we have been looking at the coastal, but we have plans to do field work deeper in the ocean, the open ocean.
Very interesting. I think when you talked about, or when you mentioned some of the viral signatures of the giant viruses, I believe you've done those in a number of different unusual environments too, have you not?
I would not say a number, maybe a few. Yeah, I mean, maybe one or two or three. Yes, we have done that. Recently, we have published a paper that looked at signatures of these viruses in this deep sea brine pool. We have collaboration with the Marine Geoscience Department in our school, and they have collected samples from this Gulf of Aqaba brine pools. So we were curious about if there are giant viruses active in these brine pools. These are deep ocean, extreme environments. They're highly saline, and not much growth happens there in the pool. So to our surprise, we have found active giant virus signatures around the edge of the pool where the growth of the microbes is the highest. And I believe we have also seen giant virus signatures right in the middle of the pool. So these things actually are thriving in these extreme environments. And previously, there has been a couple of papers that talked about identifying these viruses in extreme environments like hydrothermal vents, and also in the Yellowstone National Park. So these microbial mats in the Yellowstone National Park, these mats seem to have algal growth, and the algae has integrated giant viruses or genes of giant viruses in their genomes. So the idea here is that the giant viruses are helping transferring crucial metabolic genes in this algae. So these viruses are contributing a lot to the microbial processes across diverse ecosystem. Yeah, Mark.
I was going to say, I mean, in one, and please forgive me if this is a naive comment, but when you mention that some of these giant viruses appear to have metabolic genes, one possibility is that these are hitchhikers and they aren't necessary for the genome of the giant virus per se, although I understand, because I've seen things similar even with bacteriophages, that they might be important for the maturation of the virus. But I do wonder how much of this is just acting as a shuttle between various organisms in the mat of like sharing genetic information. That was a big interest to me back from the days of talking to Carl Woese about that. So I'm very interested in this.
No, I think totally agree with you on the sense that viruses are traditionally known as gene transfer, to some degree, agents of gene transfer among their hosts. So during the replication, a virus can accidentally pick up a host gene, and that can be transferred to a different host or different bacterial lineage during infection process. So the process is called horizontal gene transfer, as you know. So viruses are actually acting as vectors of gene transfer between these. What we see in giant viruses, though, is that a lot of these metabolic genes. So if I give you an example, if you take, for example, succinate dehydrogenase, which is a crucial gene in the central carbon metabolism pathway. And if you take the giant virus version of that particular gene, and if you build a phylogenetic tree of these genes, along with all the genes from other organisms like humans, other eukaryotes, and bacteria, so you see that the giant virus versions, they form a tight cluster, which is very separate from the cellular version. So what it tells you that these genes in the giant viruses have been persisting in their genome for, I don't know how to throw numbers, like evolutionary time scale could be millions of years. So these genes, these viruses have been carrying, and they have been maintaining in their genome for probably millions of years, many of these genes. So that kind of goes against the idea that these are temporary agents being shuttled across organisms by giant viruses.
No, I mean, that's exactly something that I'd wondered about, because if you look at a number of these viruses and they're maintaining this particular gene and it or genes related to it, and they're not being altered mutationally, they are being used for something. And that's the take-home message. I mean, I like to think of it as, I have this expression, Darwinowing, where like natural selection kind of wears away at things, and if they aren't necessary, they get worn down, and you get like the remains of genes. But that's not what's happening here. So this central, I hate to say virome, but I'm going to say it anyway. The central virome of these giant viruses, it isn't that there's a bunch of accessory information necessarily, but there's certainly a core that cannot change.
Yes. I mean, like you said, it's almost like that evolution is almost cellular. Like they have a core, genes that are like, they're central to their existence, the genomic structure. And they do have genes that are lineage specific. Like they are like those genes, you can say they are more accessory, and they might be shed off over time. They might lose some of those genes and gain new genes. But in that process, they also maintain a large core. Like
Mhm.
in each family of the giant viruses, there might be certain kind of metabolic genes that all of the viruses have, and they maintain it. They don't lose it. But there are accessory genes they can gain and lose, just like a bacteria does.
Right.
A bacterium does. So I think in many ways the evolution of these viruses kind of takes the route that we expect, the model that we actually have for bacteria, for example. So it's pretty interesting to me.
So how do you go about studying this?
Well, so studying the how these genes are being gained or lost or maintained over evolutionary time scale, there are phylogenomic methods that you can do. And essentially what we do is we reconstruct genomes of individual giant virus population using the DNA directly from the environment. We sequence DNA, we go to the field, we collect the water and sequence everything in that water. So that gives us viral signatures, bacterial signatures, and protist signatures. In the lab, we have developed pipelines that can actually identify and separate out the giant virus DNA pieces from the other DNA pieces in the same sample. And using those DNA, we can reconstruct near complete genomes of individual giant virus lineages. And once we have the genomes, we can annotate them to identify specific proteins of interest. It could be a metabolic gene. It could be any other type of gene. And taking a particular class of metabolic gene, we can build phylogenetic tree to see the evolutionary history of those genes. And that gives us a lot of information about if these are ancient metabolic genes that are being maintained for millions of years in giant viruses, or are these recently picked up from their host. So, if they're picked up from the host recently, those genes will actually mix up on the tree. They will have like a mixed cluster with the other eukaryotes. So these are one of the ways to understand the evolutionary history of metabolism in giant viruses. So that's just an example.
Sure. So what I like about this is that you have, and I dislike the term that people like to use in silico studies, meaning that you're using computers to analyze the relatedness of DNA sequences.
Right.
So there is some of that going on in your group. And in addition to that, there are folks actually working at the bench with laboratory techniques. And I actually like how those two things come together.
And I do agree with you. I do agree with you on that we are definitely working on both sides of the, in silico or in field and at bench. We're trying to cover the ground on all sides. But I do believe that there is a lot of, I don't know, like an impression, more like a negative connotation, I would say, around the idea of bioinformatics or in silico work. But one thing that I feel like as a scientist, and I think you will appreciate that too, is really we are driven by questions and curiosity. There are two things, right? I mean, you have fundamental biological questions you want to answer, and you are curious about certain things, so you want to explore a little bit. So there's more exploratory science, and there's a lot of hypothesis-driven, question-driven science. So I think traditionally bioinformatics has been attributed more towards exploration-driven science, or more like a tool of generating hypotheses. But I do believe that like every other means of generating data in science, in silico approaches are also a mean to that end. Like we generate data, but what questions you can answer from that data, that really depends on your interest and your, as a scientist, what questions you are trying to answer. And I believe that bioinformatics can be extremely helpful answering those questions if properly used and properly, you have to really do the quality control and make sure that you are doing not the wrong thing, right? Because informatic approaches, a single command, if it is wrong, if you're using a different parameter, that can lead to this disaster downstream, right? So training the students, and one of the things that I try to do with the students, I always remind them to double check, to manually check part of their data. So what they're getting as a result is actually validated and actually valid. So it's not like, they're making a big claim based on a wrong, like a command or a parameter in their informatic tool.
And that's
yes.
I was going to say that's really no different than a laboratory experiment where you must do the controls. And
Exactly.
I do want to not defend, but I do want to expand upon something that some folks in the past in particular, you're right, have kind of, I'm not going to say look down, but they've looked askance, maybe that's a better term for it, at what I'm going to call, please forgive me, just structural genomics, where you're comparing things. But the thing is, there are many times where that's the only way to come up with a hypothesis. For example, if you can't grow the organism in question, you've only found it through reconstructing it from a metagenome. All of these things can prevent you from using more traditional techniques. What I like about what you're doing is you've married the two in your laboratory group. So folks that come out of your lab have skills in both directions.
That's my goal. I eventually want to have my PhD students some experience in field and some healthy amount of experience on the bench, and also I want them to be trained in the informatics, because one way to think about it also a job prospect of the students. A lot of people nowadays don't really want to stay in the academia for a long haul. They want to explore other options, industry and stuff. So I want to train them in multiple angles so that they're well equipped for different types of jobs, and like they have more options to choose from. I have been trained in both sides, actually all three sides, field throughout my PhD and postdoc, and I want to like transfer that philosophy to my students too when I train.
Well, I think that your students are obviously quite lucky because you have a wonderful system to look at. I'm wondering if you might be able to take the listeners and viewers through a recent experiment that you found of interest in your laboratory.
Yes. So recently we have been looking into this phenomena, which a large part of my research has been during my postdoc, is looking into this more intimate interaction between giant viruses and their host. The whole idea of viruses is that they kill their host. The traditional outlook or traditional, perspective is that viruses kill. They're the agents of death. We know that very well from our own collective experience over the last few years. But another thing that remains historically underexplored is that viruses can be actually quite useful for their host. Over the evolutionary time, a lot of the host cells have adopted viral sequences. They have taken the viral sequences and used it to their own benefit. So we study this phenomena. It's called endogenization. So we're looking at endogenous viruses. One thing that recently we have found is that there is this old study. It was a very old, it's older than me actually. It's a 42 years old scientific study where a scientist have found that a healthy culture of an algae, it produces viruses. So it's more like a persistent infection of viruses that when the scientists looked at the cells under microscope, they found that these are pretty large virus particles are being produced. But the culture is not dead. It's doing fine. So one of my just exploration, one of the adventures I was doing, just reading literature, looking at old papers about this viral history, giant viruses. I found this paper on mesostigma. It's an algae. It's called mesostigma. And 40 years old paper. There's some really primitive, I would not say primitive, that sounds bad, but really beautiful, but really old transmission electron microscopy pictures of this algae. So what I noticed that these are actually pretty big viral particles. So long story short, recently we have been looking at the genome of that algae. The genome has been published to our advantage. A really good quality genome of that particular algae has been published. So the question I had is does the virus culture, the genome, have any signature that can actually provide me the answer of where are these viruses coming from? why are they not killing the host? Why are they persisting with the host in this peaceful coexistence? How is that happening? Does the genome of the host have any answer? So we started looking into the genome, and to our surprise, we have found hundreds of copies of integrated viruses in this particular algal genome. And we found that these elements, which seems to be giant viruses, for several hundred kilobase pair long, they're transcriptionally active. That means they're expressing their genes highly. So, which kind of provides you an answer to a 40-year-old question, like where are these viruses coming from? They are coming from within the cell, potentially. The cells have adopted these viruses, and we don't know why. There has to be a benefit to the cell to actually having this genome in their, having this viral genome adopted in their own genome and actively expressing these elements. So there has to be some benefit, but it does provide the answer that these viruses are endogenous viruses that the host actually have adopted and made their own. So another way you can think about an algae that actually actively produce virus but doesn't die. So that's I can say that's what it is.
No, and this is absolutely wonderful, because on the one hand, trained as a microbial geneticist with bacteria as I am, it sounds like lysogeny to me when you get a bacteriophage that becomes part of the host. And there are many cases of, for lack of a better term, what we call phage conversion, where the bacteriophage is carrying a particular gene of benefit to the bacterium. Right. And there's lots of evidence of genomes picking up viruses from nature that become part of them. Heck, the reason that we're placental mammals is a retrovirus in us. And I'll put a link to that in the show notes. Yeah. So what you're saying here is that we tend to think, and this is really important for listeners and viewers, I talk about how we're fairly narcissistic and that we're most interested in how organisms interact with us. So thus, we think of bacteria as causing trouble. We think of viruses as causing disease. And we're ignoring the cases that are kind of like meh, or in some cases beneficial. And so now you have a great model system of an alga that appears to have like the intact, well, I shouldn't say intact, it may have all the genes stayed together from the giant virus, or have they distributed?
Well, interestingly, there are elements in them that have all the genes together, several hundred kilobase pairs long, and they do have the necessary genes for capsid production or any other genes like genome packaging that all you expect from a virus, and they do have those genes. Yes.
So, I mean, it's a wonderful model system outside the ones that we're used to. Say, looking at bacteria, say, looking at certain plants. And now we have an algal system.
Yes. One quick thing is that for bacteria, lysogeny, or the fact that the viruses can integrate in the bacterial genome and stay hidden or latent, has been studied quite a bit. When it comes to eukaryotic single cells, which we call protists, and as you know, the protists are actually highly diverse, and most of the eukaryotic diversity is not in humans or mammals. It's in the protists, actually, in nature. And when it comes to protists, the lysogeny or this latent infection has not been really explored that much. It remains very underexplored, and I feel like there's a lot to learn, and I'm very excited about this system that I have.
No, I think that that's wonderful. And it must be kind of a challenge, because as soon as anyone hears the term virus, they think of negative impacts. But I know that you've given talks where you've referred to these giant viruses as the gentle giants. And I think that's a good way.
No, You're funny that you just mentioned that, because I think I came up with the term a long time ago, and I did use that in a talk. And I do believe that the more we learn about these interactions, it really seemed to be that giant viruses have these two distinct lifestyle. One is where they're actually these savage killers, right? They're destroying algal blooms in the ocean, huge blooms. They're infecting the cells in the bloom and killing, collapsing the entire bloom. And then you have this other side of them, which is very underexplored, but it seems to be extremely common in nature. I mean, in my postdoctoral work, I think that's when it all started for me. Is I was looking into these genomes of different chlorophyte algae, basically green algae. And I found that 30% of the sequenced algal genomes have almost near complete giant viruses integrated in them. So that was a huge shock to me. It seems like this particular type of lifestyle, this gentle giant kind of lifestyle, seems to be extremely common for giant viruses. So I'm very excited to explore that a little more, you know.
I mean, this is why I was so excited to have a giant virus expert, which you now are. I'm going to call you that.
I don't know if I'm an expert. I think none of us are giant virus expert in the world. Maybe there is a few, but there's so much unknown, you know?
Right. And so that brings me to another question. Where do you think things are going in your laboratory moving forward? What's really exciting to you?
Well, when it comes to virus host interactions, I think we are really on the verge of some substantial discovery or substantial scientific insights within the next few years. And one of the biggest things that in the lab that we are planning to do is really explore, as I spoke before, really establish. Well, I mean, it's already established in nature, but really understand the roles of these viruses in the global biogeochemistry, and how does over time, as the climate change, gets more intense over time, we want to understand how that impact these viruses and their interaction with their host. Do they become more relevant as the ocean worms, or do they become less relevant? Because we know they're important, but are there particular group of giant viruses that are really, really important and really, really need to be focused on to understand the oceanic biogeochemistry better? That's one thing I want to keep working on. And then the second thing is this more gentle giant kind of aspect. And I feel like this is huge. There's a huge treasure trove of unanswered question as it comes to this other side of the story. And I'm very excited to explore that. I believe, if I look back 10 years, we had what we know now, we did not know 10 times. I mean, we know 10 times less than what we know now. So I think over the next 10 years, the field will move more towards cultivating novel giant viruses and novel hosts, so we can reach consensus about their metabolism, about this hidden lifestyle, those things. And I'm very excited to be part of that, because I know that there will be more and more giant virus host system. People will start culturing them. And eventually, imagine that thousands of giant virus host systems are available, and how much insight those systems can provide us. We can really understand how do they integrate and how do they stay hidden in the host for so long. Why do the host maintain these giant viruses in their system? What is the benefit of this evolutionary benefit? And I feel like this field has the potential to change the pervasive view that we have about viruses in general. That viruses are probably not as bad as we think they are. I mean, some people might really get angry with what happened, but from a long evolutionary point of view, to maintain the diversity of life or maintain the living system as it is, they are probably very, very crucial. And we'll probably have more pieces to fit in that puzzle as we go.
I think it's a really important thing to keep in mind that, I mean, I'll give you an example. When I did my graduate work, I happened to do a rotation through the laboratory of Dr. Alan Campbell, who was one of the people who came up with the episome model about how phage lambda entered the genome of E. coli. And this is at a time that sequencing was very challenging, which is amusing for me to say. But the take-home lesson is there were 6 or 7 what he called defective prophages. That is to say, there were, and I called them the rusting evolutionary hulks of previous prophages, integrated viruses. We know that 18 to 20% of our genomes are derived from a certain RNA virus that's been lost to time.
Right.
The ALU1 sequence, for example. We know there are retroviruses, endogenous retroviruses all through us that are not causing disease. So as you say, we tend to put our focus on the negative aspects of things, and I understand where that comes from when you're trying to reduce human suffering. But it doesn't it does not give a context for what viruses are really about in the natural world. And I would like to remind you of one thing that and it's an ancient aphorism in science, where stage 1 of research is that's a silly idea, you shouldn't look at it. Stage 2 is it might be true under certain circumstances, but it's not of general interest. Stage 3 is I said it was a good idea all along. And one of my research students came up with stage 4, which was, and I came up with the idea first. And I've seen it plenty of times. And so what I'm here to say is that you may be on the cusp of watching one of these paradigm shifts that we're just talking about. I mean, there have been lots of people trying to look at positive aspects of certain types of viruses for some time. But this is something that's amenable to research and also interesting ecologically, as you mentioned.
Right. Yes. And I believe that, as we have, this is all really valid points. I don't think that nothing really undermines the importance of reducing human suffering and finding ways to cure or, prevent virus infections or pandemics like this. That's not good for us. But human life also is dependent. Our life is dependent on the biosphere. The biosphere has so much diversity. And a huge part of the diversity we cannot see. So I am excited to really understand how viruses actually contribute or help maintain that unseen diversity, the unseen biosphere, right? So, I do collaborate with the folks in the medical school here. So I am working on understanding human viruses and their roles a little bit, over the next few years. That's another thing I want to do is slowly, incorporate some of that component in my research, that I can actually start looking into the environmental distribution of some pathogenic viruses and how to actually better monitor them, better forecast them, things like that. So, I think personally, say viruses are good, that will be just half truth, right? They can also be bad, depending on how you look at it and how you actually suffer it. So as a scientist, yes. As a scientist, I want to answer those questions, and I'm curious. And my research is largely driven by curiosity, and that has been historically. So we'll see what it gets. How it kind of leads me to the next question.
Despite the fact that we've talked about not all viruses are bad, I wanted to ask a question that surely is on everybody's mind. I know that there are some types of giant viruses that affect amoebae, and that made me think about macrophages in humans even. Is there any evidence of giant viruses interacting with human cells?
Yes. One thing that I want to mention, that there has been a couple of studies, although I'm not aware of any follow-up studies on those, that have shown that viruses of amoebae, the giant viruses, can impact macrophages. I believe that study did show that they can replicate within macrophages. I could be actually very wrong. But there is a paper that talks about giant virus and macrophage interactions. I could try to find that paper and share with you guys.
If you like,
yes.
but there doesn't appear to be any strong evidence now of any negative effects.
So far, no. One thing that we know is that viruses do appear, they infect the amoebae that cause keratitis in the eyes.
Oh.
Now, it's not a direct interaction with the human cell or human organ, but it does impact the pathogen. So that part is known.
That's interesting because we're after all of us are a series of ecological niches, aren't we? When you come right down to it.
For the virus, obviously. We'll see.
I think this has been just really fantastic. And, as we start to kind of run a little bit toward the end of our session today,
Yes.
I thought that I might ask you this do you have any advice for people? I should say in this podcast, we've talked about many paths to the microbial sciences, and you've actually alluded to many paths after getting a PhD in the microbial sciences.
Yes.
So because you're closer to all of that, do you have any advice for the budding microbiologists out there?
That's a tough question to answer because, like so many paths. Everyone has a different way of looking at things and different goals in their lives. And but I would say that one thing that, and this is like a cliche nowadays, but it's so true. I would not say it's cliche yet. A lot of people don't realize it, is that, one thing that one of my colleagues told me that science is a long haul. So don't focus on like be super productive all the time, but focus on your well-being, because that will lead to productivity. If you are happy, if you are healthy, I guess it goes to the people who are like more like early career scientists, not who are looking for alternative careers. But if I want to talk about that, people really, I think we really should focus on well-being and focus on the well-being of the trainees, because a happy lab is a productive lab, which I have learned. And yes, and if I am not, like healthy, I'm not mentally there. It's kind of funny because, there was a time when I was extremely anxious about things. And I was thinking that lots of my good ideas are coming from that place of anxiety. But then I look back and I realized that that's not true. What happened is that I was thinking that I'm producing a lot of good ideas because I'm always thinking about these ideas, right? But when I'm more calm and quiet, I actually come up with better ideas. So we can do better.
I
yes.
recently saw something, and I think it's from the journal Nature, just a one-pager, and when I find it, I'll put it in the show notes, about how we all need time to think.
Yes.
And I know for a fact, as a new professor, you're discovering your time is like, forgive how I put this, it's like juggling chainsaws. You have a million things that you have to keep track of all the time, and you can't let any of them drop. And I understand that. And even when you're a more entropically challenged professor, as I am, there's still a gazillion things that need doing. And it's important to try to find a way to set aside time to just think. And it's really hard.
Yes. Because one of the biggest thing that scientists have is that they don't think that thinking is working. But, most of our ideas, most of things that we work on for years come from one illuminating thought or a chain of thoughts. Really, that really leads. And it took me a long time to appreciate that when I'm thinking, I'm working. When I'm thinking, I'm writing. People say, write. I'm like, what is writing? Writing is thinking. If I'm not happy about my ideas, I can't write it. So I have to think about it. So I think some of these things are not really talked about in academia that much. And it has been talked about is like, oh, you understand it now, I understand it now, so we don't talk about it anymore. But for the young researchers, it's extremely important to actually provide those insights and validate that what they are doing is not wrong. That's how things are done in science. Like I was thinking for a long time that today I came to lab and I have been just juggling random papers and coming up with ideas. Did I do any work? I feel bad. Did I do any work? But realistically, I came up with a good idea that probably that one of my PhD students is working on for several months now. So must be work, right? So, yeah.
And as we've discussed, there are different ways to do everything. There's no one best path, and I keep trying to convince people of this. There's lots of different ways. It's a moving target, but what really motivates folks like you and folks like me is the enthusiasm that we have for it.
Yes, I think that one of my advisors, she tells people all the time that you really have to love it. If you want to stay in the academia for a long haul, you find your place of passion in the research that you do, because that will keep you going. You know.
I got really misunderstood recently about this. There's been a lot of talk about work-life balance, and I agree
Yes.
with that. Just because you work 80 hours a week doesn't mean that you're accomplishing anything, and it's certainly not great for you physically or mentally.
Right.
But what I said once is I said that the real mark of a scientist is when they are excited to go in and look in the incubator, if it's that kind of research, to see what happens next. And that got interpreted as 80 hours a week, which is not at all what I meant. What I meant is the feeling of excitement, saying, I want to know what happens. And that's what I look for in students. That's how I've always felt. And I'm thinking that's how you feel as well.
Yes, I do. Personally, I am excited about my topic. I have been fortunate to work on a topic that is exciting, at least to me and many other scientists. But I do think that the work-life balance and the enthusiasm of a scientist, they can go together. When I think about work-life balance, to me, it's more about self-care. Like, you take care of yourself.
Mm-hmm.
You protect some time to take care of your family. You spend quality time with your partner, and if you have kids, with the kids. It does not take away from the quality of research you do. Because when you have, even if you are doing 9 to 5, taking it as a 9 to 5, 9 to 5 is 8 hours a day. It's a lot of time. And if you are focused that 9 to 5, you are working on your research, I think it's possible to do. It's taking time off has helped me a lot, actually, because what happens is that eventually you crash. If you keep going, you will crash for a week, and then that makes up for that lost time that you did not really take care of yourself. So I feel like, what's the point of it?
I think we talk a lot how it's important to be kind to others. It also means you should be kind to yourself.
Yes, and I think mentoring, one of the biggest thing that as a new early career scientist could be that you have to also train students. And that part can be, because you are dealing with different people. It's not one person you're dealing with. Several people, they have different expectations from what they want to get out of their research. Like this experience, PhD experience or graduate school experience. Everyone has different expectations. So, my job is to make sure that they reach the expectation and exceed it, but they don't break themselves. Because, in the end, what I have realized that breaking yourself, eventually, when we talk about long haul, it doesn't help anybody.
No.
If a student is sick, they need time off, because they're here for 5 years. They're not here for the month. So if they do not do that work this week, they will eventually get it done next week, because they are here for something they want to do. It's not like so mentoring. I want to remind myself all the time that you instill enthusiasm in a student by being kind. When you're kind, eventually they will come through. They will feel better, the health will get better, and they will want to do that research. Right now, they need some time off. Right now, then it's so. Not considering them as like, and I don't want to say this thing, it sounds bad, like expendable commodity or something. Consider them as partners, as of your team. That they're here for the long run. They're not here for 2 months or something.
And this is a great place to end our session today, because that's exactly the key. That it's a collaborative effort. That's certainly how research is done these days. How you do yours. Monir, I wanted to thank you so much for being on this podcast today. I have learned a lot, and I'm hoping our listeners and viewers will enjoy it as much as I have. So I want to wish you and yours, your research students, family, friends, the very best.
Well, Mark, I really enjoyed. I mean, I love to talk about giant viruses. I love to talk about research. And this is a great venue, and you are such an enthusiastic host. I mean, I think I have said things that I have not said in months, like did not discuss lots of ideas. But in this short time, in this 50 minutes or 1 hour, we talked so much about giant viruses. And I would love opportunities like this again to actually, talk about giant viruses to this big audience that you have, and I appreciate the audience that will be watching this show. And I hope they got excited about giant viruses. And not all viruses are bad.
Right. Thanks again.
Right. Yes, thank you very much, Mark.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with fabulous links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Mohammad Moniruzzaman is part of the Rosenstiel School of Marine, Atmospheric, and Earth Science at the University of Miami in Florida. Many thanks, as always, to David Renata for superb editing, and Reber Clark for the wonderfully quirky music that makes me smile each time. I hope that you've enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.